So the second video for chapter 20 is capillary exchange, and let's take a look. Okay, so this is how materials are going to be exchanged between the blood in the capillary and the tissues that the capillary is in, basically. And there's three ways for capillary exchange to happen. The first is diffusion. You learned about diffusion in chapter 4, so that's where I pulled this picture from. Diffusion is just the movement of something down its concentration gradient. So here you see diffusion of oxygen going from a high concentration to a low concentration, or carbon dioxide going the other direction, but that's still going from a high to a low concentration gradient. So that's what we mean by down a concentration gradient, high to low. Your respiratory gases are going to diffuse, whether it's in the capillaries of your lungs or the capillaries of your systemic tissues. Hormones diffuse, for the most part at least, from a high concentration to a low concentration, and waste products can diffuse, again, for the most part. Another way for things to be exchanged at the capillary level is vesicular transport. So endocytosis and exocytosis were also discussed in chapter 4. You should remember these pictures. Endocytosis is bringing materials into your cells, and exocytosis is out of or exiting the cell. But it's using these little vesicles, and they either merge with the cell membrane and then spit the contents out, or you could kind of go this in reverse. Maybe it's wrapping around some substances to bring materials in. So exocytosis or endocytosis, depending on which way the materials are moving into or out of the cell. And fatty acids are oftentimes transported this way because they're such long molecules. The other example of capillary exchange is bulk flow, and that's what we're going to focus on here in this chapter. So this is the movement of substances down a pressure gradient, not the concentration gradient that you are more familiar with. And examples here are fluid and small solutes. So those are a couple different examples. All right, I know this is scary. Trust me, it's crazy. I get it. We're not going to do the math types of problems here. I'm going to walk you through what you need to know. Let's just look at the basics right this second. This is an arteriole with blood coming from the heart, but this is a small artery, right? We just learned that. And this is our capillary. It's simplistic here. You're not seeing the sphincters and the true capillaries. We've just minimized all of that to make this a more simple view. So blood's going to come through the capillary bed, and then stuff is happening as it moves through. That's what we're going to be focusing on in this section. And then the blood keeps on traveling and goes back towards the heart. So you're in a venule, which would then go to your bigger veins and eventually get to the heart. The two main processes that are happening here are filtration and reabsorption. And definitely know what's in those little boxes there. I'm going to explain a little bit of all of this down here, but I'm not going to give you numbers and make you do math. Instead, you need to know the concepts and why this happens. What pressure is high and low, and why are things being forced out of the blood vessel here and into the blood vessel here. All right, so looking at this, there's two main processes going on, like I just said. You've got your filtration and your reabsorption. So filtration is what you see over here on the left-hand side of this diagram. Filtration means it's the movement of substances out of the capillary at the left-hand side of that diagram, which is the arterial end. over here. And that's going to be due to hydrostatic pressure. And you see that in the little red box there, because hydrostatic pressure is greater than osmotic pressure. And now you're going, what the heck is hydrostatic pressure? Give me a minute. And then on the other half of the diagram, you see over here, this is reabsorption, so the alternate process. And it's what's happening at the venous end of the capillary bed. And what's happening over there is you're bringing fluid back into the blood vessel. You don't want it all to escape. You got to bring some back in. And this happens because of the other pressure, and that's your colloid osmotic pressure. You should remember from the blood chapter that blood is sometimes referred to as a colloid because of large molecules like proteins that are in the blood. And that's what we're talking about, the presence of proteins being in the blood and in high numbers is going to increase your colloid osmotic pressure. When they're kind of more diluted, you'd have lower colloid osmotic pressure. All right, so let's take a look at both of these. The direction of the movement is going to depend on a net pressure difference between two different and opposing forces. So these two opposing forces are that hydrostatic pressure and the osmotic pressure that we were just talking about. Net pressure difference means an overall difference. Anytime you see net, it's like overall. So what's happening here is the first type of pressure is that hydrostatic pressure. I remember this because hydro makes me think of water. And this is the fluid pressure, not the colloid pressure or the protein pressure. So this is the pressure that the fluid is causing, like from the fluid pushing on the vessel wall. But there's two ways to push on the vessel wall, and that's where it gets a little tricky, and we're not going to do the math, but the fluid, you've got fluid inside the vessel, that's in your blood, right? Your plasma. But you also have fluid, interstitial fluid, that's outside of the vessel. So you'll see these little subscripts for B for blood and IF for interstitial fluid. HP stands for hydrostatic pressure. So we'll run through some of these examples in a minute, but looking down here, hydrostatic pressure, that's your net. Well, to get the net, they do some math, and the math is coming from the difference between these two pressures. So do you have more pressure in the blood, or do you have more pressure outside of the blood in the interstitial fluid? You might want to stop and rewind and listen to that again. The other pressure is the osmotic pressure, so the colloid osmotic pressure. So this is the pull of water into the tissue based on, like I said a second ago, the protein concentration that's in the tissue. So you're going to have inside is the blood colloid osmotic pressure, so how many proteins are in the blood tissue, because blood is a tissue, right? And then how many proteins and what's the pressure there in the interstitial fluid. So that's where you'll see net colloid osmotic pressure. And again, it's going to be the difference between what's in the blood and what's in the interstitial fluid. So then you end up with net filtration pressure. And that's what you see over here. And I haven't been pointing these things out, but everything down here you have on the venous end as well. So you do the math on the left and you do the math on the right, even though we're not going to do the math. That's what's making things flow, is all these comparisons on the arterial end and on the venous end. So NFP is net filtration pressure. You do not need to memorize this formula, but let's just look at it. In words, you should understand what it means. So this is your difference between your hydrostatic pressure in the blood compared to the interstitial fluid. Subtract from that your colloid osmotic pressure difference from the blood and the interstitial fluid. And so that's your net hydrostatic pressure and your net colloid osmotic pressure. And that's what you see here. The net hydrostatic pressure minus the net colloid osmotic pressure is the NFP, which is the net filtration pressure. And it's the same thing over here, but the numbers are going to be different. So here's the deal. Here's the pattern. Before I tell you the pattern, let's answer these questions here. Looking at the diagram, which pressure is the highest at the arterial end? There's 4 pressures. So you've got net hydrostatic pressure here, net colloid pressure, net hydrostatic here, net colloid here. So looking at those, let me get a different color pen. Here we go. This one is highest on the arterial end. And over here, the opposite, the colloid osmotic pressure is highest on the venule end. So what's going to happen is your hydrostatic pressure in the blood, and I've chopped this off a little bit. Let me see if I can bring this up. There we go. Let's see, where am I? Your hydrostatic pressure in the blood. So it's 35 here. Can't write on it there. Anyway, and it is 16 here. It drops as the blood moves through the capillary. And the COPs, the colloid osmotic pressures, are pretty much going to stay the same because we're not usually going to be moving the proteins through. It's usually the fluid that's going to move out or move in, like you see here. So fluid forced out of the blood vessel, fluid drawn into the blood vessel. So what's happening here is when you do this math, which we're not going to do, but when you do that math, if you get a positive number, meaning if your hydrostatic pressure is greater than your colloid osmotic pressure, that's when filtration happens. And that's always going to be the case at the arterial end. If you get a negative answer, if your colloid osmotic pressure is, let's see, you get a negative number. So if that is greater than your hydrostatic pressure, that's going to give you a negative number, and that's where absorption, or it's called reabsorption, is going to take place. So that's always going to be on the venous end. So filtration is always on the arterial end, and absorption or reabsorption is always on the venous end. Always. But it's relative to these pressures. Let's see, what else do you need to know? Here's just another way of looking at it. So, well, let's back it up a second. Hold on. Your fluid is going to be forced out at the blood vessel, at the arterial end, and that is filtration because of that higher hydrostatic pressure compared to the colloid osmotic pressure. So it's going to push some fluid out, but your proteins are going to stay, and then they'll end up being more concentrated down here because you lost some of the fluid. And so then through osmosis, you're going to bring some fluid back in because your osmotic pressure is greater down there. So this is just another way of looking at that. The hydrostatic pressure, great here compared to out here, and that is sending fluid out from a high to a low pressure scenario. And so that's going to be filtration. And this is the artery end. And at the venule end, you're going to have your So water leaves the vessel, solids stay in, their concentration rises. And so then over here on the venule end of the capillary bed, as the concentration rises, it's going to draw this water back in through that hydrostatic pressure, because now we've got higher pressure on the outside compared to the inside. So back in we go using the pressure gradient. So for the water, according to the pressure, wants to come back in. So that's absorption or reabsorption. And that always takes place on the venule end. Okay, what happens to the leftover fluid? Some of your capillaries are really only going to absorb usually about 85% of the fluid that was filtrated out or filtered out. So that means there's about 15% of that fluid that came out of your arterial end that's in all this interstitial fluid. Where does it go? Well, the answer is here. These little green tubes are part of your lymphatic system, and these are lymphatic capillaries. They take up the remaining 15%. And when we talk about the lymphatic system, we'll learn more about this, but the water flows into these little one-way street openings, and these are like close-ended vessels. It's not attached to anything, and you soak up the fluid from the interstitial fluid and send it towards your lymphatic organs. And we'll talk more about your lymphatic vessels and lymphatic organs coming up in the lymphatic system chapter. But that's another reason why you have to move around a lot. Water retention. If you don't move around, the fluid just kind of pools up in these lymphatic capillaries. So you need to move around to keep it moving and going away from your feet, for example, and your ankles. So more on that later. There are a couple of really good videos to watch. This one especially. That top one's a little more detailed and might be a little too much for you, but definitely watch the second one if nothing else to review that fluid movement in the capillaries. All right, on to the next video.